Chapter 8, Recombinant DNA Technology. In this chapter, there are 3 subtopics which are 8.1 Recombinant DNA Technology, 8.2 Method in Gene Cloning, 8.3 Application of Recombinant DNA Technology. At the end of this video, you should be able to define recombinant DNA technology. You should also be able to define and explain the 5 tools used in recombinant DNA technology, which are: first, target DNA, also known as gene of interest; second, restriction enzyme; third, DNA cloning vector; fourth, host cell; and the last one is modifying enzyme such as DNA ligase.
What is actually gene technology? It is the various techniques for sequencing and manipulating gene DNA. Manipulating genes means modifying and recombinant DNA to produce desired products such as proteins or animals and plants with desirable traits. One of the main gene technology is recombinant DNA technology. Recombinant DNA technology is a technique that joins 2 DNA molecules from different sources to produce new genetic recombination. It is commonly used in various fields in science such as medicine, agriculture, and industry.
There are 5 tools needed for recombinant DNA technology. The first one is target DNA, also known as a gene of interest. In this gene, selected to be cloned. And the second is restriction enzyme, which is used to cut DNA into fragments. The next tool is DNA cloning vector. Its function to carry the target gene into the host cell. The following tool is host cell. It allows the cloning vector to replicate such bacteria cells. The last tool is modifying enzyme, which is function to join DNA fragment together such as a DNA ligase.
Okay, in this video, you should be able to describe the gene of interest and the characteristic of host cell. As seen in this figure, genes of interest is a fragment of DNA which is part of a larger DNA strand that is in target cell. For example, the genes of interest for insulin is located in the pancreas cell. In order to replicate the target gene, the host cell is needed. It works by carries cloning vectors that contain the target gene, also known as recombinant DNA. Host cell is an organism that has been used to receive a recombinant DNA for cloning purposes, such as E. coli and yeast. There are 4 characteristics for an organism to be a host cell. First, it must be able to accept the recombinant DNA through the transformation process. Second, it must be able to maintain the structure of the recombinant DNA from one generation to another. Third, it must be able to amplify the gene product from recombinant DNA. Lastly, it must be able to express the clone gene.
In this video, you should be able to explain restriction enzyme and example of enzyme that produce sticky ends. You should also be able to explain modifying enzyme and its function such as DNA ligase and Taq polymerase. Restriction enzyme is naturally found in certain bacteria that are resistant to bacteriophage infection. It is function as an enzyme which that recognize a specific DNA sequence called recognition site, and then it will cut at a specific point on the recognition site.
There are various types of restriction enzyme. Different restriction enzyme recognize and cut a different specific restriction enzyme. This produce DNA fragment which sticky end or blunt end. The resisting site is a sequence that is consists of the same 4 to 8 nucleotides on both DNA strand but arranged in opposite direction. 5 prime A G C T T 3 prime reads, while the opposite side is 3 prime T T C G A 5 prime. The sequence of resisting site has palindromic sequence. Palindrome means the base sequence of one strand reads the same as in the opposite direction of complementary strand of DNA. For example, as shown in figure here, the function of palindromic sequence is to act as a recognition site for resisting enzyme and then cut at the resisting point on both DNA strand.
Restriction enzyme is divided into two main types based on the technique of cutting, which are either staggered cut or straight cut. When staggered cut is used to cut DNA fragments, it will form sticky ends of DNA fragments. While if the DNA fragment is cut straight through, it produces the blunt end of the DNA fragments. The table shows the example of restriction enzymes and the organism that produce the respective enzyme. So let's look at the first enzyme, EcoR1, produced by Escherichia coli. So at the restriction point, G and A, there will be a cut at both strands. Thus, this will produce sticky ends. Another example of sticky end is BAMH1 enzyme. Okay, so here the restriction point will be between G and G on both strands. While in enzyme SMAL1, it will produce blunt end. Why? Because it will cut between the C and G at the restriction sequence. How is sticky end produced?
We take our restriction enzyme EcoR1 as an example. This enzyme recognizes palindromic specific nucleotide sequences, that is GAA, TTC, and cut the DNA. EcoR1 cuts a double-stranded DNA and produces DNA fragments with sticky end. The hanging nucleotide of the sticky ends are TTAA and AATT. Thus, two DNA fragments with complementary sticky ends are formed. How is blunt end produced?
The best example that can be used is resisting enzyme for SMAL1. The enzyme recognizes palindromic specific nucleotide sequence, that is CCC, GGG, and cut the DNA. SMAL1 cut the phosphodiester bond between the nucleotide between C and G for both strands. Since the cut at the same place in the both strands, the two strand is completely cut through it. Two DNA fragment can with blunt end with no complementary base are formed.
Other than restriction enzyme, modifying enzymes are also an important tools in DNA recombinant technology. The modifying enzyme is used in modifying the DNA strand. One type of modifying enzyme is DNA ligase. It functions in joining two fragments DNA. It catalyzes the formation of phosphodiester bond during the insertion of target genes into open plasmid. DNA ligase join the target gene and plasmid to produce recombinant DNA. Another modifying enzyme is Taq polymerase. Thermostable DNA polymerase, named after the thermophilic bacterium Thermos aquaticus, and is frequently used in polymerase chain reaction or PCR.
Okay, by the end of this video, you should be able to explain the characteristics of plasmid as a cloning vector and as an expression vector. Why is cloning vector an important DNA recombinant tool? Well, because it is an agent DNA molecule that carries gene of interest into a host cell. The gene of interest will then replicate in the host cell. So there are 4 examples of cloning vector that are commonly found, such as plasmids, bacteriophages, cosmids, and YAC, which is yeast artificial chromosomes. And the most commonly used DNA cloning vector are plasmids. So what are plasmid? It is a small, circular, double-stranded DNA molecule found in certain types of bacteria. And plasmids are separated from the larger portion of the DNA and can carry up to 10 to 15,000 base pairs. And plasmid can replicate independently of the rest of the DNA. So the picture shows how plasmid looks under the microscope.
There are 6 important characteristics of plasmid as DNA vector. The first one, it has the ability to accept gene of interest, also known as the donor DNA or target gene. Second, plasmid contains an origin of replication, aka also known as the origin, to initiate the replication freely in the host cell. Plasmid can also have the ability to express the clone gene, meaning the gene can undergo transcription process and translation process. Another characteristic of the plasmid is that it contains a selectable marker genes for screening, such as gene for ampicillin resistance and also LAC-Z gene. The plasmid can also has the ability to transfer foreign gene to the host cell. Lastly, the plasmid has multiple cloning sites. This region contains several enzyme restriction sites. Thus, this allows more diverse gene of interest to be inserted.